Day 41 of growing our microbial sandbox by adding random stuff and checking it under the microscope every day. I decided to leave the sandbox outdoors to give it some fresh air and sunlight. Let's check the microscope. Bacteria are still the dominant population, followed by some teardrop-shaped protozoa. But the number of protozoa is still quite low. I spotted some new algae. It's segmented and looks a bit like a tiny nunchaku. I also found a new ciliate. It was climbing around a thread of algae, which made it look like it was doing a pole dance. Honestly, pretty good show. Time for an experiment. One of you suggested observing different layers of the sandbox. We usually look at the surface, so today, let's check the middle layer. It turns out there's not much difference. Still lots of bacteria and a few protozoa. Now let's go all the way to the bottom. Most debris has sunk there, so it's probably the dirtiest part. Surprisingly, even though there are tons of bacteria, I couldn't find a single protozoan, but that actually makes sense. Most protozoa love oxygen, so they prefer areas near the surface where oxygen is more available. Let's add more stuff today. I found an oval stone with some algae growing on it. Hopefully it enriches the sandbox. Also, baby oil. Oh no. As soon as I added it, the whole sandbox emulsified and turned milky. Not a good sign. I really hope the microorganisms can survive in this new environment. Let's see what grows tomorrow. Day 42 of growing our microbial sandbox by adding random stuff and checking it under the microscope every day. After adding baby oil, the sandbox now is completely milky. Let's check the microscope. The sandbox today is emptier than ever before. I cannot see anything moving in low magnification. Let's dive in. Under high magnification, I can still see bacteria swimming around. However, compared to previous days, the number of them is much less than before. And also, I cannot see any ciliate. Yeast cells can also be seen. I also realized that there are many circular microorganisms moving around. I don't know what exactly they are, but they don't seem like ciliates. Maybe some kinds of algae. Anyway, I noticed that some want me to add things that might be harmful to the sandbox, and some don't want. Also, the outcome after adding stuff is hard to predict. When the sandbox is destroyed, it needs time to recover. Therefore, I figured out a solution. I will take a portion of the sandbox into a glass tube as a backup before adding new stuff, so that when the sandbox is destroyed again, I can dilute it and add the backup solution back to the sandbox to revive it. Okay, let's add more stuff today. I want more neutral ingredients and nutrients. Let's go with more leaves. Chicken broth. It is full of nutrients and proteins which are essential for bacteria to grow. Chocolates. Sugar is also an essential source. That's all for today. Let's see what grows tomorrow. Day 43 of growing our microbial sandbox by adding random stuff and checking it under the microscope every day. The whole bottle looks like vegetable soup. Seems neutral and attractive to microorganisms. Let's check the microscope. I found some algae. It is Tetradesmus, a common algae that is found in fresh water and soil. Different numbers of cells tend to attach to each other. I don't know what this drunk microorganism is. It just kept rotating and seemed like it would never stop. Do you remember our old friend, Vorticella? A bell-shaped ciliate. These three just anchored themselves to the same point and hunted for their prey. Overall, protozoa do reappear. But the number is still too low. Okay, let's do an experiment. One of you asked me to culture bacteria on Agar. I prepared a brain-heart infusion agar, a common agar used for general bacteria culture. Let's see what will grow tomorrow. Let's add more stuff. Applesauce. It is one of my favorite sauces. Hope they like it. Caffeine. I just added some coffee beans. Will it energize them and keep them awake? Let's see what grows tomorrow. See you then. Day 44 of growing our microbial sandbox by adding random stuff and checking it under the microscope every day. We did so many things to save the sandbox. Hopefully more microorganisms have returned. Let's check the microscope. Look, I spotted a giant moving ball. It doesn't swim, but I can see cells moving inside. I'm happy to see that more ciliates have appeared in the sandbox. As for the dominant species, these round microorganisms are everywhere. Each one has a tiny flagellum flapping around. Instead of swimming freely, they just shiver in place or move short distances. They're similar in size to bacteria. Remember the bacterial culture we started yesterday? Now there are many bacterial colonies growing on the agar. Based on their shape and color, most of them look like gram-negative rod bacteria. To confirm what bacteria are present, I'll need a few days for further testing. Please be patient. Let's add more stuff. Rusted metal and a battery. Okay, I've got a rusted battery. Hope the chemicals inside don't destroy everything. A crusted insect. I found a dead carpenter bee. Could there be microorganisms or parasites inside? Green tea. It's healthy for us. But what about microorganisms? Let's see what grows tomorrow. See you then. Day 45 of growing our microbial sandbox by adding random stuff and checking it under the microscope every day. I see oil droplets floating on the surface. Let's check the microscope. The large bubbles are actually oil droplets. They're easy to spot under low magnification. The liquid looks a bit viscous too. There are still bacteria swimming around. Not as many as before, but still moving. They seem slower than usual. Maybe the thick liquid or inhibitory chemicals are slowing them down. I found a strange organism that moves like it's constantly shaking its head. Also spotted a euplotes. These guys use their cilia like tiny legs to walk on surfaces. And here's a microorganism that looks like a halteria, but its movement is slightly different, so I'm not 100% sure. Overall, the microbe count is still low. We need to work harder to revive this ecosystem. Let's add more stuff. Root beer. I've gotten this request so many times and I finally got some. Let's put it under the microscope. Air bubbles are everywhere. They're actually carbon dioxide bubbles. It's mesmerizing to watch them grow bigger over time. A slice of lemon. Hope the citric acid doesn't wipe out everything. And some moss. Another popular request. Maybe it'll bring in new microorganisms. Let's see what grows tomorrow. See you then. Day 46 of growing our microbial sandbox by adding random stuff and checking it under the microscope every day. The leaves look like they're starting to decay. Let's check the microscope. I found a giant amoeba. It's so big that you can even see it clearly under 100 times magnification. It moves really fast and I didn't even have to speed up the clip to show its movement. This one seems different from the previous amoeba, which needed 600 times to observe. I also spotted a ciliate that looks like a shell. I've never seen this one before. It swims around like a floating leaf. Wait, is that you, Obito? Sorry, I'm a Naruto fan. Overall, there still aren't many microorganisms in the sandbox, but I did catch one in the middle of reproducing. Look at this, it's about to divide itself. Let's add more stuff, some bacteria from my previous culture. Okay, let's add a bit. And just out of curiosity, let's put some under the microscope. You can see tons of bacteria in different shapes. Some are moving, some are not. But remember, not moving doesn't always mean they're dead. Many species can't swim because they don't have flagella, the structure that helps bacteria move. A spoonful of sugar, a classic energy source, and some watermelon. It's full of fructose too. Let's see what grows tomorrow. See you then. Day 47 of growing our microbial sandbox by adding random stuff and checking it under the microscope every day. I'm really happy to see more and more people leaving their requests. Stay patient. I will get to yours one by one. Let's check the microscope. You won't believe it. I found one of my favorite microorganisms, Ficus. It's a type of algae that moves by twisting its body in a helical motion. It has a red spot that helps it detect light intensity, allowing it to swim toward brighter areas. Its cell is flattened, with a flagellum at the head. I don't know what's going on, but it seems like more and more algae species are showing up. The one moving in the background is a diatom, which glides using a kind of sliding motion. I can still see a few ciliates, but not many. Look, I can clearly see a long thread of algae inside one of them. You are so voracious. Now let's check the comments. One of you suggested letting the sandbox rest for a day to allow more growth. I think that's a good idea. Let's give it a break today. Today, we're doing something fun. A comparative experiment. I prepared some items you suggested. Red Bull, Pepsi Cola, ordinary honey, and Manuka honey. I'll take 1 milliliter of each and mix it with 19 milliliters of sandbox content, making a 1 to 20 dilution in separate test tubes. Tomorrow, we'll see which one boosts microbial growth the most. We'll add the winner back into the sandbox. Day 48 of growing our microbial sandbox by adding random stuff and checking it under the microscope every day. Yesterday, we did not add anything. It seems the culture is not very turbid. Let us check the microscope. Not bad. I can see more ciliates swimming around. However, compared to previous days, their numbers are still low. I found an alga with a red spot that can detect light intensity. Maybe it is a modal species, though I did not see it move. I also spotted a dying ciliate. You can see its cellular contents leaking out. That is the cycle of life. Anyway, do you remember the experiment we did yesterday? Let us check the results. Let us start with the tube containing sandbox content mixed with Red Bull. I do not see any protozoa, but there are a moderate number of bacteria. In the tube with Pepsi Cola, the result is similar. No protozoa, but many bacteria. In the tube with honey, I spotted a few ciliates along with many bacteria. And finally, the tube with Manuka honey. Also, no protozoa, but many bacteria. Manuka honey is known for its antibacterial properties, but the bacteria are still surviving. We will add the tube with regular honey back to the sandbox. Next, Red 40, a type of food dye. It just adds a little color to the sandbox without killing anything. And finally, a comment printed on a piece of paper. A weird request? Let us find out the results tomorrow. See you then. Day 49 of growing our microbial sandbox by adding random stuff and checking it under the microscope every day. There's a guy inside the sandbox staring at me. Creepy. Anyway, let us check the microscope. I'm seeing more and more ciliates growing. But interestingly, only one species seems to be thriving now. I can barely find any others. The scene reminds me of Nemo swimming through coral. But these coral-like structures are actually biofilm. They are communities of bacteria held together by a sticky extracellular matrix. It helps them interact and protects them from harmful substances. Look at this pink ciliate. It is dividing. Its shape is kind of like a strawberry. I'm not sure if it is blepharisma because the shape looks a bit different. All right, time to add new stuff. Baking yeast. We already have some natural yeast in the sandbox, but a little extra will not hurt, I think. Before adding it, let us suspend it in water and take a look under the microscope. Beautiful. Millions of oval yeast cells floating around like glitter in a snow globe. Okay, let us pour them into the sandbox. Next up, mushrooms. They come with a bit of soil, so we might get some multicellular microorganisms in the mix. Here's an idea. Let us freeze them and see what survives. I'm putting a sample into a tube and freezing it overnight. We will check the results tomorrow. See you then. Day 50 of growing our microbial sandbox by adding random stuff and checking it under the microscope every day. I can't believe we are already halfway to 100 days. Time flies. Should I start a new project after reaching day 100? Anyway, let us check the microscope first. It has been a long time since I saw this many ciliates. I hope it stays like this, though I do wish for more variety in species. The sandbox is now packed with yeast cells. In case you are curious, the scientific name of baking yeast is Saccharomyces cerevisiae. It is considered harmless to humans, although extremely rare infections have been reported. The ciliates are moving through the yeast like kids playing on a playground filled with toys. The number of yeast cells is definitely more than what I added yesterday. They seem to be reproducing rapidly. Remember we froze a sample of the sandbox yesterday? Let's thaw it and take a look under the microscope. I cannot see anything moving. Ciliates clearly did not survive. Freezing damages their cell membranes. There are some yeast cells, which makes sense since we added them yesterday. I can also see many bacteria. While most are not actively moving, I'm confident some survived. Freezing kills some yeast and bacteria, but not all. Their thick cell walls help them remain intact through the freeze and thaw process. Now let's add some new items. First, egg yolk. It is rich in nutrients and microorganisms will definitely love it. Next, an antibiotic. I'm adding just a small amount to see how it affects the sandbox. By the way, can you guess what antibiotic it is based on its color? We will check the results tomorrow. See you then.